We should be going live now. Fantastic. So that worked. It's been a while since we did the CNS talk live. So welcome everyone to the first CNS talk of the season. And I'm very happy that we have Andras Jakab with us today from the University of Zurich. He did his PhD, correct me if I get anything wrong, his PhD in biomedical and imaging sciences in Hungary and in Zurich already, Switzerland.
Yes.
Yeah. Then he was a visiting researcher for computer vision in Zurich before moving to beautiful Vienna during his Marie Curie to work on computational imaging and then returned to Zurich in 2016, where he is now a professor at a children's hospital leading his own research team on fetal and infant imaging using MRI. And you just moved into a swanky new building, I saw. Are you
Oh,
part?
yes, that's correct. It's very, yes, very fancy. Yes,
Have
thank
you
you.
settled in yet?
Yes, but the researchers are not in a so fancy building. It's more like the clinicians are in this wooden building. The researchers are in a different block, but it's nice. It's
Hate to
very
say it, but that's quite typical.
right. Thank you, Stephanie, for the introduction. Let me share my screen and then I can start with this. So, hello, everyone. Today I'm going to talk about fetal MR, and also I thought I bring also neonatal MR to the topic because you cannot really disentangle fetal and neonatal development. It's more like a continuum. So I'm leading this research group, which is called Fetal Infant Imaging Group, and we are part of the Center for MR Research. And we also maintain a lot of collaborations with basic neuroscientists as well as engineers and clinicians. Now, you are all brain enthusiasts, so I'm not going to talk too much about that, but still I thought it's important to bring a few interesting facts about development to you to understand why we study this. So the brain develops from a cluster of cells to a functioning human brain, which is a remarkable journey. It starts in the third gestational week in a human, where most women don't know that they're pregnant yet. So the neural tissues develop at that time. It's a mouse neural crest, but in humans, it might look exactly the same at this age. And it develops into a functioning brain in 37 weeks, starting from here. Now, just a few interesting facts. Every minute, there are 250,000 new neurons born in this period. 1 million new synapses are established every second in gestation, and this including the glia. So the total number of neurons are 80 billion, as you probably know, depending on how people count it. And I found a paper that says that the capacity is 2,500 terabytes. I'm a bit skeptical how they came up with this, but this is still, a remarkable number when we think of it. Now, what is important, and maybe not everyone knows that at birth in humans, all the major neuroanatomical pathways, the bundles, the axons are already established, and all of our neurons exist. So from that time, it's more like myelination and actually a loss of neurons that happens, at least in the human brain. And the maximum number of synapses is reached around 2 years of age, approximately, and then it also decreases during development. Now, you can imagine that genetic mutations, environmental, maternal factors, any kind of secondary damage affecting this process can have consequences for brain development. Now, looking at it from the more neuroscientific and also anatomical perspective, brain development is remarkably interesting during the second half of gestation when it develops from this smooth, small brain into something that already resembles an adult brain um, as the gyri and sulci are concerned. So in this process, there's a lot going on. If we have a look at the cross-sectional images of the cortical layer, subcortical layers. There's growth of the brain. It grows twice, threefold in size. There's the whole cortical folding process happening in this 20 weeks of time. All the connections, at least these pioneering and then more immature connections, are established at this time. It's fully unconnected here, and it's connected at this stage. The laminae of the cortex, different layers, form, and functional specialization also emerges. So we know that there are areas already in the newborn brain defined by their functional and structural connectivity. Looking at the function, what we know from other studies, at this stage, mid-gestation, there's something called a sublate, which is a collection of premature neurons. They show spontaneous, unprovoked electrical activity. So they do something that gets transmitted to the developing cortical plate, which is important for the formation of the first synapses. In the newborn brain, we know from some studies that did resting state fMRI, there are networks already in the newborn and also a prematurely born brain. And some of them resemble that of a postnatal or adult brain. Just disappear. Yeah. But also one is a very interesting study. Maybe you like it because of your group interest. So it's a study where they looked at the cries of a typical German newborn and a typical French newborn, and they found that the accent, so this basically the tonality of the cry of these infants is different, kind of assuming there's already some sort of learning or perception ongoing in utero and in terms of language production, or at least cry production. So this is what a newborn can do, and all of this functional development takes place in 20 weeks. Now, why study early human brain development? So I like this picture. It's like an IKEA furniture assembly chart. If something is wrong in the brain because of a disease in a young child or in an infant, what we have to do is to read the building instructions again, because there's something wrong in that, and there's something we did not do correctly if we use the analogy of a furniture. Now, it's more complicated than that, but basically why is interesting to study that? I think it's important because it helps us understand human specific brain development. It helps us understand what makes us human. Also to study in a way higher cognition, at least the development of the substrates of higher cognition. And it also helps us improve the diagnosis, treatment, and patient care in diseases that interfere with this development. Now, in my talk, I'm going to talk about techniques for analyzing fetal and infant brain development. This is one of the focus of our group. Also, I brought a few studies in normal brain development, looking more like connectivity and function. And importantly, because we are affiliated with a hospital, we have a lot of clinical studies to look at pathological brain development. So to first, what allows us to quantify brain development? Typically, ultrasound is used in prenatal diagnostics. In our field, we prefer MR because of its superior ability to depict the brain. Now, what is a typical MR? So in a typical fetal MR, we acquire single-shot spinnacle images in one plane like this. And as you see, it's not a perfect scan because the fetus is moving actually a little bit, even in between the slices, because it takes a couple of seconds, 1-2 seconds to acquire one slice of these high-resolution 3 Tesla T2 spinnacle images. The same from axial plane. And as you see, the fetus is moving, so it also allows us to do some time-resolved imaging of the heart, but also on other kinds of MR, you see the whole fetal movement here. And there's the possibility to do additional, more advanced sequences. Here I cite some own works, but of course, this is not just our group. A couple of other groups are doing it. So diffusion tensor imaging, in the human fetal brain to do tryptography, as well as some sequences that are important for the clinicians, like intravoxel incoherent motion diffusion imaging, where we could capture the perfusion here in a lung lesion in a fetus, which is correlated with a neonatal malformation in the same fetus after birth. Um, but these are all challenging. So one of the challenges is subject motion and motion-related MR artifacts, which need an improvement of these artifacts, as well as the improvement of image resolution is necessary. Quantification is challenging because of the acquisition itself, but also for other reasons. We don't have a lot of ground truth models for fetuses. There's data scarcity generally, there are rare conditions, and there's, as you have seen, tremendous anatomical changes happening. And for the more complex acquisitions, many of these challenges are even more enhanced. Right, so here the aim is to implement and develop novel tools for the quantification of brain development in health and disease. So let me give you a few examples what problems we have to tackle. One is movement of the fetus, and this is quite a specific problem. So put this into perspective. In an MR experiment, let's say we acquire images in one direction, like this, if it's a two-dimensional scan. Now, the fetus lies here. It's just a drawing. It's not a real fetus. It's not that big. We acquire one slice of this fetus here, but then as we acquire the next slice, the fetus might have already moved. We acquire the next slice, it has moved, moved, moved. So as you see, the representation of anatomy is imperfect. This is how it looks like from the scanner. And this is how it looks like from the perspective of the head of the fetus. This is an image from UCL. What is happening is that it's no longer parallel anatomical sampling, but rather oblique slices are acquired for the brain. Now, from the image analysis perspective, it's a problem that can be optimized. So each of these individual slices can be co-registered with each other iteratively and interpolated to form a gradually more and more sharp and consistently looking image based on an optimization process. So this is performing super resolution and motion correction at the same time. Also, we typically oversample the fetus, meaning that we acquire stacks in different directions, which allows for oversampling and allows for outlier removal of the data. Now, after image processing, this is what we end up with. These are high-resolution isotropic images, 0.5 millimeter isotropic of the fetus, different stages of development, as you see here. And this technique is applicable to T2-weighted diffusion, DTI, as well as other sequences where we can acquire them quite fast. This is from Eleanor Oos from King's. This is for the whole fetal body with deformable registration. And this allows us to do three-dimensional segmentation of structures, not just the brain, but also the whole fetal body and placenta can be segmented. Now, I mentioned there are other modalities many of you are familiar with. People have been trying to adapt them to fetal imaging. So diffusion tensor imaging relies on the diffusion phenomenon, and in the neural tissue, it's very useful because much of it is anisotropic, and we are trying to measure the directionality of this anisotropy. Now, the fetal brain is completely unmyelinated, but we notice that the diffusion is still anisotropic because the cell membranes of axons, of neurons and other cells are still restricting the diffusion. This is by the axons, but also transient structures I'm going to show. This is very special for the human brain, for the fetal brain, sorry. And super resolution as well as the scattered data interpolation can also be used for these sequences. Now, this is an example of a fetal brain, and this is a cross-section of this diffusion tryptography that was done here. You see, this is pretty much similar, except for the fact that there's other tissue surrounding the fetal brain causing some additional problems. And what we can see in such an experiment is quite tricky because at this stage of development, it's not just the axons, but also other structures. So what are these other structures that we see? This is a 9 Tesla post-mortem high-resolution 100 micron isotropic resolution image of a human brain at mid-gestation. And what we could do with probabilistic tryptography in this fetal brain is to show the radial glia. So these are glial cell axons, and also the migrational, the tangential migrational pathways. So these are not really functioning um, connections, or not axons mediating functioning connections, but these are transient structures that we see with DTI in a fetal brain. Now, when we do tryptography in slightly older brains, what we notice is that most of the pathways are visible. So the corticospinal tract in blue, you see callosal fibers in red, as well as in green, the AP running SLF, ILF, and so on, ILOF. This paper by the London group applied the super resolution technique to the fetal DTI. I think this is already part of the DHCP data release. Now, we had a collaboration with our colleagues from CHUV in Lausanne, and what Hamza Kebiri did is very interesting. He used an autoencoder to learn how to correct DTI images, and he has learned it from preterm DTI images in the DHCP cohort and applied it on fetal. And the goal here is to better improve the resolution, especially in the off-slice direction with this autoencoder. If you're interested, I encourage you to have a look at this paper. I'm just giving you an example that fetal DTI is feasible, but we need some advanced approaches to be able to use it. Talking about techniques, and that's the last acquisition slides I have, is fetal functional MR, so bold MR. There have been a couple of publications trying to attempt that, and clearly one problem is motion. But motion is also quite specific because we have a time-resolved sequence. So in a structural MR image, as I have shown you, we do slice-to-volume recon. We are not interested in time. In a typical fMRI experiment in adults, what you do is a volume-to-volume reconstruction. So each volume, each image frame is registered or motion corrected, if you will. In a fetal setting, there's a combination of the two. So we first need to do slice-to-volume correction, but also in time you need to align these together. And you can actually benefit from different stacks acquired at different time intervals. And you also have to correct for the fact that some slices are acquired in different times in interleaved or up, down, down, up, etc. mode. So this is work from the Viennese group, from Daniel Sobotka and Roxane Licandro, where they developed this slice time correction tool for fetal MR. And this is now implemented in the nifty MIG package, if any of you is interested. So this is public code. Now, once we have corrected our data, what we can do is segment the images. So what is image segmentation? It's basically cutting out an anatomical structure, cutting the outlines or revealing the outlines. This is a task for a kindergarten child, basically, to cut out something from an image. It's very easy for a human to do this, but not for a computer. So nowadays with machine learning, this is of course an expanding field, and there's things like segment everything model, where you just type cat and it segments the cats in an image. However, people tested if these are really feasible for the brain, and of course it's not. It's very, very disappointing, basically because brains are very special and you need a lot of anatomical knowledge and doctor's knowledge to be able to find meaningful structures in it. So there's still work to do. What we have done in this field are different data releases. So we made some data publicly available. as well as we organized challenges at the MICAI conference where we invited people to develop and test their machine learning algorithms for fetal brain segmentation. So here the goal was to segment the brain into these 7 structures, and we did not give testing data for the people. They submitted their algorithms, and we kind of tested it on unseen data. There's different iterations from it, a lot of papers if you're interested. What is the conclusion here is that all the algorithms you see here, all the different research groups succeeded quite well. So there's kind of a plateau that we can reach with segmentation, which is pretty disappointing, but overall the performance is not bad. It's just a little bit disappointing that you can do it fairly easily, but you cannot really improve it too much beyond a certain accuracy. Now, once we have segmented the brain into these structures, what we can do with this data, we can chart it as a function of gestational age to have an idea how these structures grow with the developing fetus. But we can also calculate things like the gyrification index. I'm going to show you in the second part of my talk how this is used. So the gyrification index is the proportion of the cortical or pial or VM surface divided by a hull or divided by this brain coverage surface to give an idea of the convolutions of the brain. Now, this is still a challenging task that we are trying to solve. So just a little bit of technique, and then I'm moving to more biological discussions. So we are trying to improve these segmentations by different strategies. One is image synthesis. So we used the NVIDIA model where you just draw these semantic maps and it synthesizes real-looking images from there. It's again a generative adversarial network, and also denoising diffusion probabilistic models. And here the idea is that we can input a simplistic drawing of the brain, a label map, and it synthesizes a quite realistically looking MR, which can be fed into a segmentation network. So one network teaches another one. What we can do with it is to simulate different anatomical variants or pathological variants of a brain to improve the available data set. We are just working on that with Misha, postdoc in my group. We can simulate different ventriculomegaly, asymmetric, other conditions in the brain, and we synthesize real-looking MR from this data. Also, an advantage is that these are not patient data, so these are synthetic, artificially modulated data sets, and therefore it can be shared more easily because they don't belong to anyone. I just talk about newborn brain. I'm sure you better understand now because it's very similar to an adult or even child imaging setting. Something is special that they're sleeping in the MR. But from that point, we acquire DTI and structural images, and this allows us to do connectomic research. I'm not going to detail that. The aim of connectomic research is to understand how connections are organized at the whole brain level, which allows us to calculate different metrics, different measurements, characterizing this organization, for example, efficiency or different modules that are formed in the brain. And more importantly, in this field, how these develop and how they are affected by diseases. So to sum up the first part of my talk, the techniques, we face a lot of challenges. We can use different techniques, especially AI, very popular these days for image quantification. Starting from the late fetal period, mapping structural connections is possible at the detail that I was showing you. And the first connectomic studies in newborns are already published, which help us understand the organizing principles of the brain. Now let's have a look what we can learn about normal development. I am not going to give you an overview of everything about development. I just brought a few interesting studies. So as we know, there's rapid development in the second half of gestation. There's axonogenesis, synaptogenesis, cortical arealization ongoing. And why we do it, why we study normal development. It helps us to identify windows of increased vulnerability in the brain, vulnerability to diseases, but also it helps us understand some human-specific aspects of development. I'm going to show you some examples for it. Now, just to put this into a perspective, once we have acquired structural images of a lot of fetuses at different gestational ages, we can establish models that describe cortical development and human brain development in general, based on normally developing fetuses. So this is data from Vienna, from Georg Lanz, his working group, where a four-dimensional cortical surface model has been established. What you see here is the emergence of primary sulci, secondary sulci, and corresponding gyri, as well as gyri in the frontal lobe. So this is a pretty interesting procedure, and it also helps us find interesting aspects in the human brain. One of the interesting aspects that has been studied for a long time is hemispheric specialization or hemispheric asymmetry. Now, it has been known that newborn brains already show this morphological asymmetry. Gregor Kasprian has described that already the fetal brain shows temporal lobe asymmetries. So the superior temporal gyrus depth is rightwards asymmetric, and it appears quite early, mostly determined by genetic programs. Now, we conducted a study where we had real longitudinal scans of the same fetus. So Sellensteger in my team acquired images of a fetus, 32nd week, and the same fetus as a newborn. Also, we had a control group with healthy infants. What we had what we could look at in addition to the main study hypothesis is to study brain asymmetry. So we could basically co-register each brain to the flipped version of its brain and characterize how the asymmetry changes from fetal to the neonatal period. You can read more about this technique in this published paper. So basically, the Jacobian determinants could be statistically tested. So this allowed us to do two different kinds of tests. I'll quickly walk you through these plots. First, to characterize what are the regions in the fetal stage that are asymmetric. What are the regions in the neonatal brain that are asymmetric in terms of macromorphology? And then what are the regions in the brain where the asymmetry changed from the fetal to the neonatal time? So what are the dynamics of this in a given individual? Now, we could use two different methods. One is to compare the fetal and a neonatal asymmetry statistically that you see here. But what we could also do is to calculate the growth rate in each individual from the fetal to the neonatal time point, and then calculate the asymmetry of the growth. So these are two complementary methods. One of the findings that were found in both methods is the temporal pole asymmetry, which appears to change its degree of asymmetry in this last part of fetal development. As you see here, so this left temporal lobe gets, let's say, more asymmetric as in a newborn compared to the fetus. What you see in the static asymmetries are quite a few known asymmetries here, the superior temporal gyrus, temporal pole. I would say this is precentral gyrus, not very accurate, and also some parietal asymmetry. In the neonatal, a leftwards asymmetry in the postcentral gyrus, as well as some subcortical and other statistically significant asymmetries were found. And something that changed the asymmetry in this test was also parietal, but also quite a few temporal areas have been dynamically changing the symmetry. So this makes us think that there is genetic programs ongoing in the brain that drives this morphology, but maybe also the environment and maybe a little bit reflecting functional specialization in the brain. A lot of theories exist why auditory and language gets asymmetric at this stage. maybe asymmetric auditory input to the fetal brain. But I'm not going to discuss that today. This is more just a observation and nothing functionally tested. So structural connectivity also develops rapidly. I'm just going to show you a few examples. What we see in a mid-gestational brain is that there isn't really gyri and there isn't really functional and structural areas characterized by divergent connectivity areas. It's more like gradients of connectivity that we can see in this brain. And as the development progresses, what we see more and more is that well-circumscribed areas in the cortex appear where the connections are more segregated. Now, this theory was formulated by Zhao and colleagues. They said that it's the structural segregation that improves or increases prenatally, so the formation of cortical areas. And then after birth, it's more the integration, so the long-range connections that interconnect these modules that are developing. Now, we haven't fully put this to a test. I just show you a glimpse into that. We analyzed the DHCP fetal release DTI data, tested how modularity changes and how global efficiency changes in the fetus. It's not exactly confirming it. Modularity maybe increases a little bit, and global efficiency seems to decrease. It's still a matter of interpretation, and maybe it starts to increase in the end, which would support this hypothesis, but I think we need more old fetuses or maybe preterm brains in addition to that. Function of the fetus, I think it's interesting to you. There's been ancient papers where they stimulated the fetus with sounds, and they found acoustic activation or activation in the brain and temporal lobe as a response. So these are, 1999, so very old studies, and probably the printers were also not very good. But there's also a reinvention of these studies where they looked at resting states. So Mariah Thomason, but also we published a few papers in this field and found that there are bilaterally connected resting state networks in the fetal brain, as well as some unilateral networks. Default mode network is not really found here. Yeah, so these are more like primordial networks. Now, what we are doing at the moment is something called FET lung study. So it's about functional development. I refer back to this study where they found that the cries of the infants are already different. So our assumption in this study with Alexis Revis Adelman and Alejandra Husser was that language development begins in utero. So our study protocol is as follows. We have an audio system put on the abdomen, and we record the voice of the mom and create different modification of that to be speech-like, non-speech-like, and to carry different pitch properties and also silent periods. And we have a sparse MR acquisition, meaning that we acquire images and then stimulate with sound so that they are not overlapping, so that the readout happens when there is no specific sound stimulation, hoping that the HRF is so prolonged that we can record the response related to these stimulations. Now, this is an ongoing study with 2 participants. I hope to publish this in the next year. Now, what happens in a pathological brain? I have 10 minutes for that. I'm going to show you 2 conditions that we have studied in the past in my group. One is congenital heart defects. So congenital heart defects are the most common congenital abnormalities affecting 1 to 2%. Here, what happens is that the development of the heart and great vessels is abnormal, and there's abnormal circulation, which affects the brain. Survival is very good because of surgery is very effective these days, but people have noticed more long-term effects on the brain because of the good survival rate of these infants. The most affected domain are executive functions, so it mostly affects higher cognition. So the aims of these studies is to find out how cerebral this maturation is linked to outcomes and how to diagnose it early on, maybe even fetally, and how to identify risk for these brain development and cognitive developmental problems. So in the brain CHD study, Celine's PhD work, we analyzed MR data in a following study protocol. We had an MR fetally of heart defect fetuses as well as control groups. We had then an MR after birth as newborns, and then the heart children had several surgeries and several brain MRs were performed, and we measured neurodevelopmental outcomes as well. As you see, we have 2 time points for not all the subjects. In this study, it's just 26 and 8 controls. But this allowed us to do a preliminary analysis, which is now being written as a paper. So they have found, or we found in this measurement that confirming the literature, that total brain volume is already reduced in neonatal and fetal stages, maybe not in the mid-gestation, and maybe this difference gets more exaggerated towards the end of gestation. Looking at the individual parts of the brain, the total volume of white matter is apparent between the congenital heart group, which is blue, and the control, which is red. But it was not very convincing. So the other parts of the brain were not that different, and the rate of growth was also not very different. In another study where one of my students worked together with a children's hospital in Shanghai, we actually looked at the gyrification index. And interestingly here, it seems that the gyrification index shows bigger differences than merely the volume differences, meaning that it might precede the volume loss. So these more qualitative changes in the gyrification process are more apparent in the fetuses with congenital heart defect. You see it in green. This is also study in progress. In another study already in newborns, we had a look if outcomes, so cognitive outcome measured with the Bailey composite score, is linked to brain volumes. And here you see the effect size. So cerebellar volume seems to be the most associated or explaining the variance, which is very interesting. We don't quite understand why. People would be expecting, let's say, frontal regions, but this is what we found and published. We were also thinking what really contributes to these volumetric differences. We know that in heart defects, it's not just the genetics, it's not just the oxygenation problem, but there's also multiple hits. And one of the hit is actually the operation itself. So the heart surgery that the children undergo. So what we did in this study, if you're interested, there's a paper from it now. We looked at the intraoperative desaturation curve and formed groups between these infants that underwent more oxygen loss, more desaturation during the operation versus those infants that did not have such desaturation events. What we found is that the lateral ventricles were much bigger in those who underwent this worse surgical outcome, so to say. And when we did a voxel-based morphometry, what we found are periventricular regions. Probably it's periventricular white matter injury and periventricular white matter loss that contributes to this brain loss in congenital heart defect. So it's multiple hit. Some of it is fetal, some of it is postnatal. For language, we published this paper a few years ago. In the same group, we had longitudinal MRs in each individual before surgery and after surgery. We had a look at the volumetric changes with VBM or DBM in this cohort and tested whether the cognitive development is explained by these growth changes. Now, this is very interesting and very significant, might not be specific to CHD, but what we found is that the growth of the left planum temporale predicts the language score at 1 year of age. So this baby language development measure is linked with the growth of this region, which is quite a fascinating finding because this time interval is really just a couple of months. Now, we did connectomic studies. I'm not going to go into details. We're approaching, I think, the end of the talk. Some of the whole brain connectomic studies confirmed the abnormalities at the global scale in infants with congenital heart defects. To sum up what these global differences are, so they have seen increased segregation or increased modularity and decreased integration in this paper. Now, if we think of the developmental model, this might speak for a less mature brain because we thought, or Jung et al. Thought that the fetal brain is more segregated compared to an infant brain. So maybe they are lacking or lagging in connectomic development. Now, this is an interesting way to analyze the data. We can put the connectomes into a two-dimensional morphospace defined by modular organization and integration. We did that with control children, congenital heart defect children, and children with other conditions like spina bifida, premature birth. And what we found is that there is probably a deviation from this normal zone of connections in CHD, but it's not that trivial. Preterm born and other newborn diseases are more deviating from this optimal organization of the brain connectome. So this is now a paper under review. And we also found some weak links to outcome scores with the small verdness. So again, the same integration captured here. I'm going to skim through this. There's another condition called spina bifida, quite common in fetuses. It's a spine problem, but it causes a lot of brain developmental problems. And these infants or these fetuses are operated fetally in Zurich. So they are operated in the womb. And we had a look at their cognitive development. So cognitive development is a late manifestation of the disease despite the surgery. Surgery improves the motor functions, but it doesn't improve the cognitive functions of these infants. So we had a look at over 100 cases, how the fetal brain growth and fetal volumetric changes are associated with the 2-year outcomes in these babies. We use some machine learning segmentation to measure the volume of different brain parts in the fetus and in the newborn, and we calculated the growth curves, the growth rate of each part of the brain. And in a preliminary statistical analysis, this is from Kelly Payet from the group, we found some correlations, quite good correlations with deep gray matter and language outcomes, as well as the motor outcomes at 2 years of age were linked with the volumetric changes of the ventricles and the cerebellum in the fetus. So this is almost like a prediction of, or at least an association, a long-range association. Now, in another study on the same cohort, we looked at thalamocortical connectivity patterns. This is published now, and what we found is that the different neurological outcomes of the babies, like what is their functional neurological lesion score, can they move independently, are they wheelchair bound, is associated with the topography of the thalamocortical connectivity as mapped by DTI and connectivity-based clustering. So to conclude these pathological studies, we found that these conditions lead to global changes in the brain, and general clinical risk factors also contribute to connectomic abnormalities. Right, and we found some links to outcomes, and probably this is influenced by the clinical picture as well, and not just the underlying congenital abnormality. So to conclude my talk, there is a link in several aspects of early development and later cognitive outcomes in newborns at risk for developmental delays, such as congenital heart defects, spina bifida, and we were able to look at these links thanks to the advanced techniques that we could use in fetuses and newborns. Many of the structure function links that we discussed are secondary in nature, so they are not primarily caused by the disease, but mostly caused by the whole trajectory of the disease and these additional hits during development. I think long-term follow-up studies are needed, and we are already conducting some of these, for example, to look at school performance, 5-year, 8 or 10-year-old cognitive development of these children. And it's also important or interesting to look at other traits, comorbidities, other conditions. I think autism, ADHD are, of course, a big topic in the field because many people assume they might have pre or perinatal origins and genetic origins as well. So with this, I'd like to thank you for your attention. In Zurich, we have some nice views, and in Switzerland. So with this, I close my talk, and I'm happy to answer questions.
Thank you so much. What a wonderful overview from the method to the healthy brain to the clinical applications. Quite impressive. Um, so before I open the discussion to the people in the audience, I also want to take this opportunity to invite the people over on YouTube to write their questions, and then I'll bring them over here. Generally speaking, I was quite impressed to see that you have quite large participation numbers. I would have expected that for fetal and early developmental studies, the numbers tend to be low because our experience is that it's very difficult to recruit. But you seem to be doing quite all right. What's the trick?
Oh, thanks. That's a compliment, but actually our rates are not that good. I think to recruit healthy fetuses or pregnant women, very difficult. So I think one
Mhm.
of the studies, one of the many studies, could recruit maybe 20 such cases in 3 years of time. So this is not a good recruitment rate. We have more success when we recruit pathological ones because then there is a clinical indication already. Sometimes it's not a very strong indication in case of congenital heart defects, because an ultrasound is probably sufficient. But at least from the ethical perspective, we are in a safer ground because there is clinical benefits from an MR scan. We approach through the regular sources like advertisements. Um, yeah, I think some people are just intrinsically motivated to contribute to science. So some of the pictures was from my daughter who also underwent a fetal MR scan in the womb. So she's just born a month ago.
Oh, congratulations. Yes, she's a keen participant in all your studies going forward.
We have.
Um, I give the floor to Muge. She has a question for you.
Hi, thank you. This was very interesting. Thank you for this talk. I was just wondering about the group analysis in, like baby fetal fMRI, for example. Are there like templates for each gestational stage, or like is it? I don't know how that would work. So.
Yeah, that's a very good point. I did not have time to go into that. There weren't many templates when I started this in 2013 in Vienna. Now there are a couple of very good developmental atlases, high resolution, super fancy atlases that you could use for spatial standardization. It still remains a challenge, so you have to consider if you would just group your subjects around an age and use a mid template, a midpoint template. In some of the cases, it's easy. So, for example, for one of the studies, we recruited 32 gestation, so 32nd gestational week fetuses. So they were the same age roughly. So there, the normalization was done for the 32nd week. In studies where we have real longitudinal, like many ages, then you cannot really do voxel level statistics, obviously.
So, like those figures that you showed, like the gestational stage on the x-axis and the other like developmental measures that you were testing. So those are from like a single fetus, or
Yes, those are
that?
different. So those are cross-sectional studies. You cannot really do in the
Mhm.
fetus many MR scans. Usually it's one. So those
Ah,
are different individuals.
okay, okay. Thank you. Yeah.
You're welcome.
Eva has a question.
Hey, thank you for the talk. It was very interesting. I have first a clarification question of the study with the different accents of crying. The stimulation for that one, I don't know if I saw it correctly, but it's under a second or
Oh, you mean the language study, the fetal language study? Yeah, so the acquisition is a sparse EPI, meaning that the readout lasts for 1 second. So that's when the image is acquired, and then nothing happens in the MR, and then there's the stimulation for 1 second. So the effective TR is 2 seconds, but the image is just acquired for 1 second.
okay, and you build this with the mother's voice, and it will be
Yes. Pre-record them.
substantially different in just 1 second.
It's not it was on the slide, but I did not talk about it. They're just syllables, so like ga 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 la 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 la. So these are very simple speech-like stimuli.
Right, right, okay. And then for the acquisition, I was wondering if there is some live motion estimation that can be done or that is in the works.
I am aware of this happening, but I'm not aware of anyone really using it for the fetus. It's mostly retrospective. So the strategy in the past 10 years has been to acquire images as fast as possible, structural, but also fMRI, and then try to correct it retrospectively with image processing. For live monitoring, I don't know. I mean, it's very difficult to monitor the fetal brain, so even with ultrasound, probably you cannot do as good as with retrospective because you don't see the whole brain movement in the womb. From like acquisition level motion correction, I think those are mostly for structural scans. So I'm aware of some techniques from Siemens, I think also Philips has, where when they do the motion correction as the image is being collected, as the case space is being sampled. But unfortunately, this is not really done for the fetus.
Right. Thank you.
I can jump in here quickly. So I was wondering, you showed that the data acquisition is isotropic 0.5 millimeter, which is obviously a beautiful resolution. But it also makes your life harder in terms of motion artifacts, I'm assuming. So the question is, why, other than we all want the highest resolution, but where is the best trade-off for your kind of work in terms of resolution and anatomical?
Yeah, so that was the resolution of the reconstructed image. So the trick here is that the original images that I've shown are good resolution in plane 0305 with a single shot, that's doable, but the off-plane resolution is 2.5 to 4 millimeters. So they're very fast slices, but we acquire 3 planes twice, maybe more times. So there are fetuses with 12 or 15 repeated MR scans. So that's the trade-off that we trade off the off-plane resolution, and then we collect all these images and then combine them into one single super resolution image. So that's the trick that many groups are now using, and it has even reached the clinical practice. As I know in London, they like it very much. We also sometimes do it for the clinicians so that it's in packs and they can have a look at this reconstructed image. Motion is kind of dealt with because the algorithm can kick out the slices which are affected by the motion. So we have another slice from which you can interpolate.
Right. And you also mentioned that you acquired them sleeping, which makes sense. I was
In
wondering,
the newborn, yes.
yeah, I was wondering, so a couple of years ago, silent MR imaging came about, which is obviously extremely exciting.
Yeah.
Is that something that's routinely used?
Yes, we are doing it. So we work with GE for some reason, and G has looping star. That's the name of a new sequence. It's completely silent for structural imaging, so you don't hear the MR, you just hear the helium pump. That's good. For both, we are running subject studies on adults. It has some noise because of it's still a
Yeah.
traverse rapidly, but it's very silent. Now, actually, we don't see a big benefit compared to the sparse acquisition in terms of the auditory response. So the sparse acquisition is silent when you present the stimulus. What you say about waking up, I think we can only do this anyway for infants between 0 and 4 weeks of age. Once they are 2, 3 months old, they don't sleep that deep. So they would wake up anyway after 10, 20 minutes. With a newborn, I think they just sleep through 40 minutes easily, and it doesn't matter if the MR is clicking or not. So they are not really woken up by the MR, they're woken up by their tummy, or they just don't want to sleep. So there it's safe. I don't know what to do with the older infants, but probably it's not the MR that's causing a problem.
Interesting. And then the study you showed about the association between different anatomical structures like the white matter or the gray matter predicting future cognitive, motor, and language skills. So the cerebellar connectivity is the most predictive
Yes.
feature for cognition, language, and motor functions?
Yeah. Yes, yes. Well, yeah, yeah, yeah. So the question is why, or what do you think? Or
More, how do you interpret it? What do you make
yeah,
of it?
yeah. It's difficult to interpret because here we deal with we deal with pathological brains.
Mhm.
So you should not jump to the conclusion that the cerebellum is responsible for these cognitive functions to the greatest extent, because we know that it's important, but not necessarily in this sense. What you could conclude from this is that probably the cerebellar volume is affected by the disease the most for some reason, or it's developmentally affected, or a general lag in development affects the cerebellar growth. So this is what I refer to as more secondary effect, and not really that, okay, the cerebellum is now the language, and then, it controls. It's more like a multiple hit. Yeah, especially in heart defects, we know that the cerebellar growth is affected. So it might be just a moderatory effect of clinical cumulative risks from the disease and development in general. Thanks for the question. This is really what we are asking ourselves, and this is very important.
Well, it's also not an outrageous association, right? Because there's more and more studies demonstrating
Yeah,
at least the
yes,
involvement of the cerebellum.
yeah. The CHD is not very surprising. It's quite a weak association. Yeah.
Thank you. Bob has a
Yeah,
question.
yeah. I just coming back to the cerebellum topic. I've seen a study recently that also showed a association between cerebellum myelination, or I'm not sure what it was, and educational attainment and cognitive abilities. But the reason why it was so strong is the authors pointed out that the other tracts, so they were looking at tracts, I believe, white matter, they were all very highly correlated with each other. And if you add cerebellum into the mix, it was not very highly correlated. But if you throw them all into a multiple regression model, then they all explain the same amount of variance, but the cerebellum, because it pops out a little bit, that it increases the amount of variance that is explained by the cerebellum. So it could be a really simple statistical artifact that also plays into this a little bit. I'm not sure if this is happening with your data.
I agree. This is a good point, and I think this could be part of what's happening, that the cerebellum is just intrinsically more variable, and also in later stages of development, it just develops a little bit more rapidly. because it's very tiny in the fetus and then it grows quite remarkably. So yeah, I can see that point, that there's just more natural variance in those measurements. Thanks.
Thanks, Bob. I can just about hear the clocks outside, so that
Oh,
means we are hitting 5 o'clock. Thank you so much for joining us today and for this wonderful overview covering a wide range of topics from the methods to the healthy brain to the clinical applications. Quite impressive, as I said. Thanks for bearing with us for all the questions that we had, and lovely to see you again. For everyone who joined us, the next talk will be Flavio Dell'Acqua, talking about high-resolution diffusion tractography imaging, and we yet have to fix the date end of the month because something came up. But I keep you posted on that one. Thanks for joining us today. Bye.
thank you. Thanks. Bye.
Bye.
Bye, thank you.